Thank you, and I'll try not to stand too far in the way of you and afternoon tea. As you can tell, I am not a PET scientist. My background is in CNS drug discovery. And I'm going to start with a very obvious point about drug discovery. There are some good things about it. They've led to some really positive impacts on people. So here we have the top 5 prescribed drugs in Australia in the 2021 financial year. We've got some statins, some reflux drugs, a hypertension drug. And if you add up the number of scripts that were prescribed, there's over 50 million there in that 1 year. It's enough for 2 for every Australian. So drug discovery does have some really big impacts on people's health, but there's some bad sides to drug discovery as well. It's a very inefficient process as it stands. So for every one of these approved drugs, there's another 4 that fail in clinical trial. And given the amount of money and time that goes into clinical trials, that's a lot of inefficiencies. And then if you take a step back to the preclinical phase, for every one that succeeds, there's 249 that fail, and even more at the phase before that. And that's inefficiencies in the process, but there's also inefficiencies in time. For every one of these drugs, it takes on average about 10 to 12 years to produce one, and over $1 billion approximately to produce that as well. And if we zoom in on what the most common cause of those inefficiencies are, this is an analysis in Nature Reviews Drug Discovery a few years ago. Over half of those failures, the majority, were from efficacy. So the drug just didn't do what it was meant to. It didn't help the disease or help the patients. And so total body PET could be something that really helps with this efficacy problem in drug discovery. So I have to acknowledge Professor Michael Cassio, who's my boss, who's given a talk on this topic before. This is a slide from his talk. Pharmaceutical executives were asked to identify over the next 3 years what area is going to play the most important role at their company in drug development. And one of the most popular answers was molecular imaging. So how could total body PET help with this efficacy problem in drug discovery? Well, I'm going to give you 3 examples, and depending on whether I get a finish sign popping up, I might give you a few more. The first is with PET microdosing. So one of the reasons that a drug can fail is that it doesn't get to the organ we want it to. And that's particularly with some of those neurology and neuropsychiatric drugs. The blood-brain barrier stops a lot of them getting into the brain. So PET microdosing is a way to have a look at whether drugs get to where we want to before going and doing a full clinical trial. You administer a very tiny, maybe 1% of the therapeutic dose of a drug. That drug's radio-labeled. Small number of people, phase 0 trial, less than 10, and you can chase that through. And so one of the areas that this has had impact is in one of the fields I'm in, which is oxytocin. Some of you may have heard of oxytocin as a pro-social molecule. It was portrayed as the panacea for all sorts of asocial disorders, social anxiety, schizophrenia. Unfortunately, though, intranasally administering oxytocin hasn't fulfilled that promise. It's been fun watching the headlines. They're like their puns in this field, watching the headlines come about as that happened. Some of the studies show effects, some of them don't. Some of them even show deleterious effects. And PET microdosing has been one of the things that's helped understand why that might be. There's a few different reasons, but this is an example of a study that's tried to answer this question. Here we have the amino acids that make up oxytocin. One of them's been radio-labeled, had an intranasal, this is a preclinical study, intranasal administration to the mouse, and you can see whilst a fair bit ends up in the olfactory bulb, not much gets into the brain proper. One of the things, this hasn't been done in humans, that's still pending, but one of the things that total body PET will help with these sort of distribution studies is we had the beautiful image from Simon earlier with the injection of the radio-labeled substance, and you can see it spread throughout the whole body. So total body PET will allow it, if it doesn't reach the organ of interest, to tell us where it does end up. If it's in the fat stores, maybe it's too lipophilic. If it's in the kidneys, it gets excreted too quickly. And then you can go back and do some more chemistry on the molecules to try and design out those liabilities. Maybe the drug does get to your organ of interest, but perhaps it doesn't actually bind to the target it's meant to, whether that be a receptor or a transporter. And so that's where target engagement studies come in. So how they work is you get some sort of PET radio tracer to, let's say, your receptor of interest. It's got to be really high affinity. It's got to be selective to just that receptor, and it has to have the right sort of pharmacokinetics to get to your organ of interest. And that's really hard to do. We've been bashing our head against a brick wall for a few years trying to find one for our particular receptor of interest. But what you can do if you do have one is inject that and then administer your drug that you're developing. If there's a big signal, PET signal, when that happens, it means that your drug isn't binding to your receptor of interest because it's not displacing the radio tracer. But if it does generate a low signal, that means that it is binding to the drug protein of interest. So you're getting target engagement. And in our journey with various different pharma companies with drug discovery, a number of them have said that their interest in acquiring academic intellectual property are much higher if they can see some target engagement through these studies. And one of the pharmas that we're working with at the moment, part of our program is to develop a new radioligand to be able to show target engagement of our drug. And this is just an example. Many of you will be aware of psychedelics and the reduced restrictions that are available now for figuring out how these work in humans. So psilocybin is one of the psychoactive ingredients in magic mushrooms. Psilocybin is known to bind to a whole bunch of different receptors, but it's not really known which ones contribute to the psychedelic effects. And so a target engagement study using a radioligand to one of the serotonin receptor subtypes was carried out, and you can see that psilocybin is able to greatly displace the binding of that radioligand, and that happens on a time scale that's approximately when those psychedelic effects kick in the self-reported questionnaires that happen. So it does seem like 5-HT2A receptors are one key receptor that psychedelics work through thanks to these target engagement studies. And there's a whole bunch of drug discovery that's happening at the moment where people are trying to design synthetic 5-HT2A agonists to use for neuropsychiatric disorder treatment. Another flavor of these type of experiments are receptor occupancy experiments. So maybe a clinical trial fails, even though the drug's getting to the organ of interest, it's binding to the right receptors, maybe not enough dose has been administered. So receptor occupancy studies allow you to find the right dose to give in clinical trials. And so that's establishing how the percentage of receptors bound associates both with dose and also the physiological effect that you want. So it's doing those target engagement studies, but administering different doses. So those of you with good eyes might be able to see down here that different amounts of, in this case, psilocin, which is another psychoactive ingredient in magic mushrooms, different subjects were given different doses, and that was correlated with the plasma concentration of psilocin. And you can see that the percentage occupancy, which is 100%, is when you just have the radioligand there. 0% is if the drug of interest has completely displaced that signal, and you can set up a dose response curve to understand how dose correlates with receptor occupancy. You can then also correlate dose with the intensity rating or some sort of physiological effect. So in this experiment, the physiological effect was self-related psychedelic effects from a psychedelic questionnaire. And so then you can put those two together and get an idea of how receptor occupancy can dictate the physiological effect. What's unknown at the moment with these psychedelics is what sort of intensity rating is needed for a therapeutic effect. But once that's known, you can then read off this curve the sort of percentage occupancy you need for your therapeutics. And so that'll be really important when all of those 5-HT2A drug discovery studies come to fruition and the clinical trials start. They'll be able to figure out exactly how much of a dose to administer in those clinical trials. And where total body PET comes in handy, again, a concept that Simon mentioned in his talk, you'll notice here again, those of you with keen eyes, that each of these different doses have been given in different people. And that leads to a lot of noise in these studies. And so with the sensitivity and the opportunity to use lower doses of the radioligands, you're able to potentially do multiple doses in the one subject and hence really reduce the noise that you see in these experiments. The points I'm going to make now have been touched on by Gemma and Matthew and Simon, but there are other opportunities for total body PET to help with drug discovery. Different drugs might work for a subset of patients, and if you can predict those with various biomarkers, then you can pre-screen your patients for clinical trials, which will increase the amount of efficacy in those trials. Biomarkers are a great way to see whether or not drugs are doing what you want to the physiology. So using something like TSVO radioligands to map inflammation over time. And with total body PET, and again, the lower dose needed, that opens up being able to maybe use multiple time points and follow people for longer across the course of the clinical trial. And as has been mentioned a number of times too, there's a real growing interest in how different bits of the body, whether it's peripheral inflammation, the gut, influence the brain. And so being able to image all of those at once and administer drugs will give us a big insight in clinical trials about the interaction between all of those systems. There are definite challenges for all of this. So in terms of the CNS, there's radioligands for over 30 different brain targets. And some of the targets have multiple radioligands, which is great. There's so many for the muscarinic acetylcholine receptor. But there are, of course, so many more proteins, receptors, transporters of interest in the brain. And so the challenge is to get radioligands that bind those. And again, as has been mentioned today, the other challenge is interpreting whether or not the signal you're getting is from the radioligand or from a radiometabolite. So to sum up, there's some big areas where drug discovery needs improving, particularly related to efficacy-related failures. And total body PET offers great opportunities for helping that out, whether that be with distribution studies and microdosing, with target engagement, with receptor occupancy, or in improving clinical trials. And congratulations to all the team for getting things to this stage. And I look forward to seeing all the great discoveries that are going to come from here. Thank you.